Hi, everyone. So I'm going to shift gears a little bit from finfish to shellfish for my talk. I just want to give a heads up that this talk is basically covering a large review that this team did here. So I have several collaborators from Dartmouth College that helped out with this that couldn't be here today. So you'll see a lot of references to something that's in review right now. So just bear with me as we go through some of the slides. So just to start out with, why are we talking about PFAS and shellfish? As you've heard from several talks today, PFAS are a widespread environmental contaminant group that accumulate into some aquatic organisms. And as was mentioned by Megan just a few talks ago, shellfish is one of these groups of seafood that we know could be a route of exposure to PFAS. However, when we actually think about what the term shellfish encompasses, it could mean different things to certain people, and it actually includes a lot of different organisms. So it could include things like bivalves. This could be oysters, clams, mussels, scallops. Could include crustaceans. Over here is a picture of one of my favorite lobster rolls. So if anyone is ever in New Hampshire, just save my information right now. I can give you a recommendation for a great place for lobster rolls. Sort of a competition everyone has up here. So, but there's also other things like certain cephalopods like squid, octopus, and certain gastropods like snails, limpets, and whelks. So all of these different organisms can count as shellfish if we're looking at dietary exposure. But as you've heard from some of the other talks, depending on an organism's lifestyle or its habits, that might impact the way it accumulates certain contaminants, including PFAS. So when we think of PFAS sources, we've heard from several talks that there's different ways that PFAS can enter the aquatic environment. And once they're in there, they could partition into the surface water column. Maybe they could be forming films at the surface, at the air-water interface, or the sediment-water interface. And we also know that there could be some absorption into sediment and also certain particulate matter. But from there, we started thinking about things like, is it bioconcentrating or is it bioaccumulating, depending on the organism? And here we sort of have a breakout of some examples of different shellfish where really depending on which organisms you're talking about, they could be undergoing different processes and could have very different concentrations and profiles of PFAS in them. So this could be a real problem when we think about epidemiological studies that are trying to assess exposure and they're simply asking, do you consume shellfish? Because there could be a huge error on that. Are you talking about predatory shellfish that maybe are accumulating higher concentrations, or maybe certain sediment-dwelling organisms that could have a completely different profile? So the goal of our work was just to try to go through and see what's the state of the science on this. How many studies have been done on this? Because we know a lot of work has been done on finfish, especially on freshwater finfish, to understand accumulation and risk to consumers. But we want to get a lay of the land of where we at with shellfish. So as far as a lot of the studies, they've pretty much been going on only for about the last 10 to 15 years, and a majority of them have been taking place in either Asian countries or in Europe. So a lot of this is dealing with coastal organisms. There's less information about freshwater animals. And a lot of these are also just part of ongoing monitoring programs. So maybe some folks here are familiar with Mussel Watch. That's a program that collects mussels from along coastlines and looks at a variety of persistent organic pollutants. And in recent years, they started including PFAS in those results. Similarly, other monitoring programs look at non-point sources of PFAS. So just thinking general coastal areas have detected PFAS in certain shellfish. And then there's smaller universe of studies that have started really focusing in on places like AFFF use sites, airports, or certain industrial sites that maybe are near coastal regions. So more and more people are starting to include shellfish studies or starting to include shellfish analysis in seafood studies, but it's still really lagging behind what we know about finfish and especially freshwater finfish. So one of the big things that we've noticed is when we look through these studies, several people have probably noticed in their different media and different areas that folks work in is if you go back about 10 years, the bee's knees was looking at PFOS and PFOA. But as time has gone on, this family and this suite of analytes that we're looking at is just exploding, and we're getting a lot more compounds that are being included, and the detection limits are really ranging. And this can be an issue, especially if we're trying to look back at historical exposures where In some cases, the detection limits are as high as 10 parts per billion, and now we're into the parts per trillion practically when we're looking at analyses. So not all of these compounds are included across every study, which makes it difficult to compare across species, across habitats. But it is pointing that there is interest in understanding where are these other compounds, what are these replacement products that are possibly being detected in aquatic organisms. So one of the things that's pretty noticeable if you go through the literature on this is that there is some variation across taxa. So these graphs are on a log scale. So even though the bars are kind of close, they're sort of still wild and all over the place. But one of the key things that does pop out is PFOS and PFOA tend to be amongst the highest concentrations in several organisms. And some of that is due to their historic use and a lot of sampling that has gone on in the past, but also that the majority of literature is really focused on these because they're the two PFAS compounds that we always keep coming back to in the research. But there are also some taxonomic groups that have not really been well studied, especially when it comes to things like, for example, cephalopods, gastropods, some of those mollusk animals, and really looking at some of the either precursor compounds or some of the sulfonic acids and really understanding what's accumulating into those. So if we even sort of zoom in onto one taxonomic group, if we look at a group like bivalves, this is some example of recent data that we're working up here in New Hampshire, where we've done some sampling of certain bivalves based on habitat types and some different feeding approaches. And one of the things that we're seeing is, a clam isn't the same as a mussel, isn't the same as an oyster. If they're sampled in similar sites, they have very different ranges of concentrations in PFAS. They also have very different profiles that make that up. And this is really raising some interesting questions for us of, are these differences due to feeding habits and differences in bioconcentration and accumulation? Is this differences in toxicokinetics, where maybe some of these organisms are better at eliminating certain PFAS than others? Or, as you can kind of see in this graph, the bars off to the side are showing known precursor compounds. And are we having differences in metabolism where, for example, maybe with blue mussels, maybe they're producing PFPEA because of breakdown of precursor products or something else? Or if there's actually PFPEA in the water column, then they're uniquely retaining this. So this is really getting to that key issue again, where, shellfish is a really complicated and loaded term when we start digging into it. And that could have implications for what we think about with exposure assessments. So when we even look at bioaccumulation factors that have been generated in the literature for some of these taxonomic groups, it's really limited to mostly bivalves and crustaceans. And when I say crustaceans, that's including things like mysid shrimp and some crustaceans that we don't typically eat. But there's a pretty wide spread on these bioaccumulation factors. So if you look across the x-axis, we go from short chain to longer chain carboxylic acids, then short chain to longer chain sulfonic acids. You can see there's a pretty wide spread across these different taxonomic groups on taking that ratio of what's in the environment to what accumulates into the organism. So there's not a lot of research on precursors. And again, that might be leading into questions of, is there biotransformation? But compared to finfish and other vertebrate animals, this is really still understudied. And that's a problem for risk assessment, but that's also a problem for when we think about ecological risk assessment or even looking at regulatory approaches to what we might set as limits in the environment. So one of the things that you could do is you could take some of this data, you could look at some of the ranges of what we see in these studies, and we can ask, okay, what does the hazard quotient look like? So the idea of a hazard quotient is it's an estimate or ratio of an exposure to some threshold for risk. So that threshold could be an EPA reference dose, it could be an ATSDR MRL. There's a lot of these different values that keep evolving, but it's asking, if you exceed this hazard quotient, if you're above 1, then there could be risk. And if it's below 1, then it's unlikely a health risk for a consumer. So again, since not a lot of epidemiology studies really tease this out, we thought we'd take a look at this review that we did of the literature and ask, with a certain group like bivalves, what do we see with those hazard quotients? And this is what we generate. So the vertical gray line that's going up is basically a hazard quotient of 1. So where you see the bars crossing it, that's where some of the ranges of the data for that compound might exceed risk for those groups. And we looked at the 50th percentile of consumers for adults, women of childbearing age, and children. And then we looked at sort of a high-end scenario of the 90th percentile of consumers. So it's a little bit busy of a graph, but it's basically this idea that if you're crossing in those high-end scenarios, there may be risk to consumers. So in several cases, we see PFOS and some of the longer chain compounds are definitely crossing for those high-end shellfish consumers. But for the more typical shellfish consumer, we're not really seeing much crossing that hazard quotient. One of the things to bear in mind, though, is a lot of these studies that we pulled from that resulted in those high-end values were from around the time of the phase out of these products. So in the early to mid 2000s and sort of going into the early 2010s. But more recently, we haven't really seen concentrations that are going past that threshold. So some of that may be because of where bivalves sit on the food chain, and they may not be accumulating that much. So for human risk, it's sort of still an open question, because maybe if we're looking at impacted sites, there still could be uncertainty to that risk. But we still have to think again about what about ecological risk, and how could this be building up into other predatory species that might be consumed? Now, one of the things I always like to do with presentations is state the things we don't know about and what are all of our shortcomings. And one of the big challenges is analytical chemistry, right? Because we are chasing a growing list of these compounds in various tissue, various media. But one thing that's important to remember is methods differ. The method for drinking water is not the same for fish tissue, is not the same for shellfish tissue, and you can get mixed results from that. A good example that we had here in New Hampshire was at an AFFF site on our coast. They did testing for shellfish, and all the samples showed up with PFPEA, a 5-chain carboxylic acid. And it caused a lot of panic and worry, because folks are saying, oh my gosh, all the shellfish have this, they're all contaminated. It caused a lot of worry in our shellfish industry. A lot of our shellfish farmers were worried what this meant. But when we sent it to EPA Office of Research and Development, they were actually able to find that it's an interferent. It's a natural fatty acid that occurs in bivalves that if someone's not looking carefully when they're doing analysis for PFAS, they'll get a false positive for PFPEA when in fact it's just a natural compound in the shellfish. So that's one of the things that we've been working on, is trying to find other groups that can help us out with this analysis, make sure that we're getting real results. But it's a thing too with not just seafood, but other media, is to bear in mind, when can we be getting these wonky results as we keep growing this family of analytes? And another challenge here for New England is a lack of capability and capacity, where we don't have a lot of labs that can do seafood analysis. If you talk to most states right now, a lot of us are shipping stuff to Vancouver to do analysis, or to some private labs. So that is an opportunity for researchers. And another thing that is really complicated is talking about risk and relative risk. And the slide is only funny if you have Disney Plus or if you really like Disney musicals. But one of the things we don't talk about in risk assessment often is this issue about risk, relative risk, and risk versus benefit. So that fun equation that I showed you earlier to look at risk and potential harm of consuming shellfish, there's no variable, there's no term in there for balancing benefit of eating seafood. So if you go to a nutritional epidemiologist and you ask them what are the benefits of seafood, they will rant and rave about the benefits of seafood and show you all kinds of graphs that quantify the benefit of consuming seafood. But for us in the realm of environmental contamination, we're usually looking at the opposite of what do all the graphs show us on the risk of consuming seafood. At this time, it's something we don't have an answer to. We don't have a way to actually balance those two when we make recommendations. And as we discover more PFAS, other emerging contaminants, and as the thresholds for these go lower and lower, there becomes a question of, for vulnerable groups or economically vulnerable groups, do we end up limiting their access to certain foods out of risk when we're not able to look at benefit and weigh that accordingly? So it's a really complicated issue. It's something that goes beyond being just a toxicologist. It gets into the issue of policy and environmental decision-making, and really something I'd encourage folks to think about. So with that, I'm happy to take any questions. I do want to acknowledge my team at my state agency, both in the Department of Environmental Services and the Department of Health and Human Services, as well as the great team that I'm working with at Dartmouth College. We're collecting a whole bunch of data on shellfish and fish and water and sediment. So hoping next year to come back and just crush everyone with 15 straight minutes of slides on PFAS data. So with that.
We are at time, so we'll move on to the next presenter.